Good afternoon. Today we are going to learn in more details about the smooth muscles. We have said that the smooth muscles, they are non-striated. So they are known as smooth muscles and they are involuntary muscles. They are usually supplied by the autonomic nerve, the sympathetic and the parasympathetic divisions. The smooth muscles can be multi-unit and unitary smooth muscles. There are gap junctions which are present in the unitary smooth muscles, and these help in transmission of impulses quickly, and they function as electrical and mechanical syncytium due to the presence of these gap junctions. We know that a typical neuromuscular junction is present, that is a type of chemical synapse which is present in the skeletal muscle, in the smooth muscle and the cardiac muscle. When the nerve terminal, it comes near the muscle ending, there are some dilations formed which are known as varicosities. And the gap between the presynaptic terminal and the muscle end plate decreases, and there are gap junctions present which helps in transmission of chemicals as well as electrical impulses. In the skeletal muscle, we have seen there are presence of Z lines, and the sarcoplasmic reticulum is very well developed. The terminal cisterns of the sarcoplasmic reticulum along with the T-tubule, which is the invagination of the subcollateral membrane, form a triad. While in the cardiac muscle, we have seen due to the placement of the difference in placement of the T-tubule, there is a structure formed that is called as dyad. That is one terminal cistern of the sarcoplasmic reticulum and one T-tubule. In the smooth muscle, you can see here there are presence of caveoli. The sarcoplasmic reticulum is also not so well developed. So for contraction in the smooth muscle, influx of calcium is usually necessary. In the smooth muscles, there are absence of Z lines. Instead, there are presence of dense bodies. There is presence of thick and thin filament, that is the actin and myosin filaments. There are gap junctions. Troponin is absent in the smooth muscle. So in this picture, you can see the varicosities in the nerve terminals, and inside is present the neurotransmitters. Now, control systems of smooth muscle, that is contraction or inhibition of smooth muscle, can be initiated by intrinsic activity of pacemaker cells, can be neurally released neurotransmitters, they can control the activity, or circulating or locally generated hormones or signaling molecules, they can control the activities. The combination of a neurotransmitter, hormone or drug with specific receptor activates contraction by increasing the level of calcium in the cell. The response of the cell depends on the concentration of the transmitters or hormones at the cell membrane and the nature of the receptor present. Hormone concentrations depend on diffusion distance, release, reuptake, and catabolism. Consequently, cells lacking close neuromuscular contacts will have a limited response to a neural activity unless they are electrically coupled. so that depolarization is transmitted from one cell to another. Now you can see the pictures A and B. A is multi-unit smooth muscles, resembles striated muscles. In that there is no electrical coupling and neural regulation is important. So there is no electrical coupling present in this multi-unit smooth muscle. In single unit smooth muscles are somewhat like cardiac muscles, and electrical activity is propagated throughout the tissue by the gap junctions. Most smooth muscles probably lie between two ends of the single and multi-unit spectrum. That is, there will be mixtures of both present. Now, let us revise about the excitation contraction coupling in the cardiac and the skeletal muscle. So, you know, in the cardiac and the skeletal muscle, the troponin-tropomyosin complex keeps the active site of the actin filament covered. So when a depolarization wave, it travels down the sarcolemmal membrane, down the T-tubule in the skeletal muscle, what happens? There is a change in the voltage, conformational change in the voltage gated calcium channel, that is the dihydropyridine receptor channel, which causes a mechanical pull in the ryanodine receptor channel, which is present in the terminal cistern of the sarcoplasmic reticulum. This causes release of calcium from the sarcoplasmic reticulum into the sarcoplasm. This is known as electromechanical coupling. What happens in cardiac muscles? When the depolarization wave travels down the T-tubule, it causes opening of the dihydropyridine receptor channel, the voltage-gated L-type of calcium channels, and as a result, calcium moves into the cell. This calcium causes opening of the ryanodine receptor channel. So, the ryanodine receptor channels open and calcium is poured from the sarcoplasmic reticulum into the sarcoplasm. So this is known as electrochemical coupling. So in skeletal muscle, it is electromechanical coupling, while in cardiac muscle, it is electrochemical coupling. Now, rest of the steps, what happens? 4 calcium bind with troponin. See, this causes the troponin-tropomyosin complex to move deeper into the groove of the actin filament. As a result, the active sites of the actin filament are now free. The myosin head has got ATPase activity in both skeletal as well as the cardiac muscles. It breaks down ATP to ADP and PI and stores the energy. Whenever the active sites of the actin filament are free, the myosin head binds with the active sites of the actin filament. Now, so what happens after that? There is a power stroke that is generated now, that is tilting of the head occurs, and this causes the myosin head to release the ADP and the PI. So now again another ATP gets attached with the myosin head. When myosin head is attached with the ATP, it loses its affinity for the active site of the actin filament. As a result, the myosin head gets detached from the active site of the actin filament. It gets detached from the active site of the actin filament. Now it again breaks down ATP to ADP and PI and moves towards the next active site of the actin filament and gets attached to it. In this way, the myosin head moves along the actin filament, pulling the actin filament along with it, causing contraction of the muscle, and the length of the sarcomere decreases. For relaxation of the muscle, what is essential? Again, energy is required. For detachment of the myosin head from the active site of the actin filament, we require ATP, and for bringing down the calcium level in the sarcoplasm, we require ATP. In the skeletal muscle, we have seen that calcium is coming from the sarcoplasmic reticulum. It is not essential to bring calcium from the extracellular fluid into the intracellular fluid because the sarcoplasmic reticulum is very well developed and contains plenty of calcium. So there is a pump, that is the calcium ATPase, sarca pump, that is a calcium ATPase pump, which pumps back calcium into the sarcoplasmic reticulum, which pumps back calcium into the sarcoplasmic reticulum, which binds with this calcium is now bound in the sarcoplasmic reticulum by proteins which are calciquestrine and calreticulin, so that the ionic level of calcium inside the sarcoplasmic reticulum remains low. This causes a buffering action so that the sarca pump can continue to act. Now, in the cardiac muscle, what happens? Some calcium has come from the ECF. So this calcium needs to be moved back to the ECF. There is a sodium calcium exchanger which is present in the sarcolemmal membrane, and this will cause calcium to move out of the cell via a secondary active transport. Now we will discuss about the smooth muscles. In the smooth muscles, there are different types of action potential. Some can be spike. You see the pictures. Some may be spike. Some may be plateau potential, spike potential, and there may be slow waves. For the slow waves, we have discussed in the last class, there are voltage-gated calcium channels. They open and there is rise in the membrane voltage, which is responsible for the action potential spike. So inside the cell, there is increase in calcium concentration. Now, this increase in calcium causes opening of calcium-dependent potassium channel. When the potassium channel opens, the membrane gradually hyperpolarizes. So, the voltage-gated calcium channels now close and internal calcium gradually decreases. After that, calcium-dependent potassium channels also close. So the membrane again starts depolarizing. This causes again opening of the voltage-gated calcium channel and the cycle gets repeated. We will again discuss about this graph. This is the relationships between the membrane potential and the generation of force in different types of smooth muscle. See the figure A first. Action potential may be generated and lead to a twitch or larger summed mechanical responses. Action potential are characteristics of single unit smooth muscle. Gap junction permit the spread of action potential throughout the tissue. Now you come to figure B. Rhythmic activity produced by slow waves that trigger action potential. The contractions are generally associated with a burst of action potential. Slow oscillations in membrane potential usually reflect the activity of electrogenic pumps in the cell membrane. Now see, tonic contractile activity may be related to the value of the membrane potential in the absence of action potentials. Graded changes in EM are common in multi-unit smooth muscles where action potentials are not generated and propagated from cell to cell. Now the last is the D, which is a pharmacomechanical coupling. Changes in force produced by addition or removal of drug or hormones that have no significant effect in the membrane potential. You can see there is no change in the membrane potential. Now we come to the contractile mechanism of the smooth muscle. So regulation of smooth muscle myosin interactions with actin by calcium stimulated phosphorylation. In the relaxed state, cross bridges are present as a high energy myosin ADP-PI complex in the presence of ATP. Attachment to actin depends on phosphorylation of cross bridge by a calcium calmodulin dependent myosin light chain kinase. Phosphorylated cross bridges cycle until they are dephosphorylated by myosin phosphatase. Cross bridge phosphorylation at a specific site on the myosin regulatory light chain requires ATP in addition to that used in each cyclic interaction with actin. So in this picture, you can see that calcium, what happens? 4 calcium ions bind to calmodulin, which is closely related to the troponin C. You have seen troponin C in the skeletal and the cardiac muscle. Here, instead of troponin C, there is presence of calmodulin. In the skeletal and the cardiac muscle, you saw 4 calcium was bound with troponin C. Here, 4 calcium, they bind with the calmodulin. The calcium calmodulin complex activates an enzyme, that is the myosin light chain kinase, which in turn phosphorylates the regulatory light chain that is associated with each neck of the dimeric myosin 2 heavy chain. So you can see it in the picture. Phosphorylation of the light chain causes a conformational change of the myosin head, which increases the angle between the head and neck domain of myosin and also increases its ATPase activity, allowing it to interact efficiently with actin and act as a molecular motor. So in smooth muscle, instead of troponin C, we can see there is presence of calmodulin. Contraction cannot begin until myosin light chain kinase increases the ATPase activity of myosin. And this is relatively slow and time dependent. In the skeletal and the cardiac muscle, the ATPase activity of myosin head is high And the cross-bridge cycling begins as soon as the tropomyosin moves out of the way. Now, in this picture, you can see covalent regulation allows 8 cross-bridge states in smooth muscle. Phosphorylation by myosin light chain kinase. You see the vertical red arrows is obligatory for cross-bridge attachment. Phosphorylated cross-bridges cycle comparatively rapidly. Dephosphorylation of a cross bridge during a cycle by a consecutively active myosin phosphatase. You can see the vertical back arrows, slow cycling rates, and produces the latch state. Calcium regulates cross bridge cycling by determining phosphorylation rates. ATP is required for both regulation, you see the vertical arrows, and cycling, the curved arrows. Again, the same picture you can see here. Two proteins, caldesmol and calponin, tonically inhibit the interaction between actin and myosin. Both these proteins, they prevent the or inhibit the interaction between actin and myosin. Caldesmol is another regulatory protein of smooth muscle that appears to act like calponin by tonically inhibiting the actin-activated ATPase activity of myosin in smooth muscle. So we have learned about two new proteins. One is calponin, another is caldesmol. Caldesmol and calponin. What are the functions of these two proteins? They tonically inhibit the interaction between actin and myosin. You can see there are different types of calcium channels are present out here in the smooth muscle. Calcium enters the cytoplasm through channels located in the caveolae. There are also, when the sarcoplasmic reticulum calcium stores become depleted, the sarcoplasmic reticulum signals by an unknown mechanism a store operative calcium channel to open, allowing calcium to enter. Calcium is also released from sarcoplasmic reticulum via calcium-induced calcium release or via IP3 activation of sarcoplasmic reticular calcium channels. So there are different ways in which the calcium level can increase in the smooth muscle. Now, let us discuss about the Rho kinase signaling in smooth muscle. A variety of agonists of G protein coupled receptors simultaneously stimulate INSP3 production and activate Rho kinase signaling. INSP3 is produced by phospholipase C mediated hydrolysis of PIP2. This we all know. INSP3 increases intracellular calcium by opening INSP3 gated calcium channels in the sarcoplasmic reticulum. You see the picture given on the side, resulting in calcium calmodulin dependent activation of myosin light chain kinase and phosphorylation of the myosin regulatory light chain and promotion of actin-myosin interaction. Activated Rho-A stimulates Rho kinase, which inhibits myosin phosphatase by phosphorylating the myosin binding subunits of the myosin phosphatase. So activation of Rho-A, what does it do? It stimulates Rho kinase, which now what does this function of this Rho kinase? Inhibits the myosin phosphatase by phosphorylating the myosin binding subunit of the myosin phosphatase. Rho-K also inhibits myosin phosphatase indirectly by phosphorylating or activating CP117 or 17 KDA inhibitor of myosin phosphatase. The net effect of Rho kinase phosphorylation is a decrease in myosin phosphatase activity. So there is decrease in myosin phosphatase activity, which results in an increased level of myosin light chain phosphorylation and hence a greater force of contraction at a given intracellular calcium. Now, in the skeletal, cardiac and the smooth muscle, you can see here that the calcium, instead of troponin C, there is presence of calmodulin. And you can see the ryanodine receptor channel, the isoforms are different for the different muscles. Even the sarcopum, some isoform, there is a difference in between them. Now, the response, we will see something about the more in details about the smooth muscle. The response to sustained or tonic stimulation is a rapid contraction followed by sustained maintenance of force with reduced cross-breed cycling rates and ATP consumption. This behavior is called the lax state, is advantageous for muscles that need to withstand a continuous external force. Example, the blood vessels, which have to withstand the blood pressure. During the latch, ATP is consumed at a much lesser rate. Example, like 1/300 times less as compared to maintain the same force in the skeletal muscle. The length tension relationships, hyperbolic velocity load relationship, power output curves, and the ability to resist imposed loads are comparable to those of skeletal muscle in smooth muscle. Shortening velocity and ATP consumption rates are very low in smooth muscle. There is another property of the smooth muscle that is unique property, that is length adaptation. That is the ability to adjust the length tension relationship when chronically stretched or shortened. This process is called the length adaptation. Length adaptation. Here you can see in this picture the length adaptation of the smooth muscle. So in the first picture A, you see both skeletal and the smooth muscle, they exhibit a bell-shaped length tension relationship. But the length tension relationship can vary in a smooth muscle. Within a short period after stretching, the smooth muscle, what happens? There is a rightward shift in the length tension relationship, such that maximal force generation occurs at a longer muscle length. Within a brief period after shortening of the muscle, there is a leftward shift in the length tension relationship. The mechanism or mechanisms underlying this length adaptation are hypothesized to reflect a change in the number of contractile units in series. See the picture C. Now we come to something about the force velocity curves. Force velocity curves for fast and slow human skeletal muscles and smooth muscles. You can see in the figure A. Now in B, you can see smooth muscles have variable force velocity relationship that are determined by the level of calcium stimulated cross-bridge phosphorylation. In C, maximal shortening velocity with no load are directly dependent on cross-bridge phosphorylation by myosin light chain kinase. Now we come to the figure D. Active force by stress rises rapidly with phosphorylation, and near maximal stress may be generated with only 20 or 30% of cross bridges in phosphorylated states. So this was about the force velocity curves. Now we will discuss something about the pharmacomechanical coupling that is seen in the smooth muscles. The smooth muscle, we have already discussed about the rho kinase pathway. So we will discuss something about the pharmacomechanical coupling. Some smooth muscles contract without action potentials. We have told there may be application of some drug, some hormone, which without a change in the membrane potential can bring about contraction in the muscle. In smooth muscle, both entry of extracellular calcium and intracellular calcium spark activate contraction. You have seen that the striated muscles, they have got T tubules. In the smooth muscle, they have got caveoli. And smooth muscle cells use three major pathways for rise in the calcium. Calcium entry through L type of calcium channels which are present in the caveoli, calcium release from the sarcoplasmic reticulum via ryanodine receptor channel and IP3R calcium channels, and calcium entry through voltage independent store operated channels. Through voltage independent store operated channel. Calcium entry via voltage gated channels. What is this? Whether the smooth muscle cells respond to graded depolarization or action potential, depolarization may produce a calcium influx through L type of calcium channels. Calcium release from the sarcoplasmic reticulum, we have seen they can be via the ryanodine receptor channels or via the inositol triphosphate activated channels. Now, calcium entry through store operated calcium channel. Depletion of sarcoplasmic reticular calcium stores causes aggregation of STIM1 monomers in the sarcoplasmic reticular membrane, thereby activating Ori channels in the plasma membrane, allowing calcium to enter the cytosol for uptake into the sarcoplasmic reticulum by sarcoplasm. So calcium entry through store-operated calcium channels. Both calcium release from the sarcoplasmic reticulum and the entry of calcium via ASOCE are voltage independent. These two mechanisms underlie the pharmacomechanical coupling, a form of excitation contraction coupling in which intracellular release of calcium is initiated by chemical activators rather than the membrane voltage changes. So, what are the mechanisms? That is, calcium release from the sarcoplasmic reticulum and the entry of calcium via the ASOCE. These are voltage independent, and these are the two mechanisms which underlie the pharmacomechanical coupling. And this is a form of excitation contraction coupling in which intracellular release of calcium is initiated by chemical activators rather than membrane voltage changes. So the membrane voltage doesn't change. Instead, there are some chemicals which causes increase in the release of intracellular calcium. A variety of drugs, excitatory neurotransmitter, hormones can induce smooth muscle contractions by this coupling mechanism independently of action potential generation. So this are not, there is no change in the membrane potential in this instance of pharmacomechanical coupling. There may be a sudden rise in the calcium level due to calcium that comes from the PCF. There is also some increase in calcium which is released from the ryanodine receptor, and this sudden rise in calcium is known as the calcium spark. So calcium can enter the cytoplasm through the channels which are present in the caveoli. Now, some agonist acting through G protein coupled receptors can lead to formation of IP3, and this IP3 can act on channels which cause release of calcium. Also, calcium entry via the channels in the caveoli activates the ryanodine receptor channels. Calcium induced, calcium released. Depletion of sarcoplasmic reticulum calcium stores causes aggregation of STIM1 monomers in the sarcoplasmic reticulum membrane, thereby activating Ori channels in the plasma membrane, allowing calcium to enter the cytosol for uptake by the sarcoplasmic reticulum by the sarca pump. Now, for relaxation of the smooth muscle, again, what we require? We require the calcium to be pumped back into the sarcoplasmic reticulum or out of the cell. There is also requirement of myosin phosphatase enzyme. And for the pumping back of calcium into the sarcoplasmic reticulum. There is again the sarca pump, and for removing calcium from inside the cell, there is the calcium ATPase pump present in the sarcolemmal membrane, as well as there is the sodium calcium exchanger. Both these can decrease the calcium level inside the cell. So we have learned about the smooth muscle, and we have discussed about the smooth muscle, cardiac muscle, skeletal muscle, their differences. So next day, we will discuss about some of the applied aspects. Thank you. Bye.